Hi there, I am Dr. Stewart, and today I'd like to give a brief overview of the small intestine. So this is an abbreviated version of a longer lecture. So for more details, please check out one of my full-length videos on the small intestine. So as a quick review, here is some major portions of the digestive system. Here we have the esophagus that carries food down into the stomach, which is drawn in green. The stomach's job is to break down a lot of that food into a substance called chyme. It's a liquid pea-like soup-like substance called chyme. That chyme is then moved into the first part of the small intestine, which is the duodenum. The duodenum then connects to the jejunum, and finally the ileum, before then it moves into the large intestine. So overall, the job of the stomach is to break these large nutrients that we ingest down into smaller pieces. Like we consume these really large proteins, these really large carbohydrates. The stomach's job is to break those into smaller pieces. Now, the job of the small intestine really is to absorb the nutrients that have been broken down. Okay, by the time those nutrients get into the small intestine, they're smaller, but they're not quite small enough to absorb. Because remember, in order for the body to absorb nutrients, sugars have to be broken down into their smallest pieces, which are monosaccharides, and proteins need to be broken down into their smallest pieces, which are individual amino acids. Okay, so the job of the small intestine is to finish breaking those nutrients down in their smallest pieces and finally absorb them into the body, right? Now, one characteristic of the small intestine that makes it effective at absorbing nutrients quickly is that it has a really large surface area. Number one is long. It's like 16 to 18 feet long, right? So it's a tube that's long. That's going to increase its surface area. But most importantly, it has some other features which dramatically increase the surface area on the inside lining of that tube. For example, if you consider, let's assume the small intestine was like a hose pipe. So it had a smooth surface on the inner wall, which is not the case. If that was true, though, if it was like a hose pipe, the surface area of our small intestine would be about the same as one of these classroom desks. Okay, so it's big, but not huge. However, due to some other features, the surface area is a lot bigger. One of those features is are called circular folds. Let's say we take a section of the small intestine and we do a slice and we open it up so we can see inside. You're going to find that there's a bunch of little wrinkles, like circular wrinkles, that are located on the inner wall of the small intestine. These wrinkles kind of wrap around the tube like this. That's going to increase the surface area right off the bat. Furthermore, if you were to zoom in on one of the surfaces of those wrinkles, you would find that they're covered with these tiny finger-like projections. So here's a zoomed-in view of what the surface of one of these circular folds look like. All these finger-like projections are called villi. These villi are small, but you can still see them. It makes the inside of the small intestine almost look fuzzy when you look at it with your naked eye. Then, if you were to zoom in on the surface of a single villus, you would find that the epithelium that makes up the lining of these villi is really unique. So here's the wall of a single villus. So we just zoomed in on this little part right here. It's going to be composed of simple columnar epithelial cells. Okay, that's what each one of these little cells represent. But the apical surface of these cells that faces the inside of the gut contains all of these tiny little finger-like waves in them. And these are distortions of the cell membrane itself that make up these epithelial cells. Each one of these tiny little finger-like distortions, those are called microvilli, and those further increase the surface area of the small intestine. When you consider all three of these things together, the microvilli, the villi, and the circular folds, the actual surface area of our small intestine is about the size of a pretty big classroom. So we've increased the surface area from the size of a desk, compared to just a hose pipe with a smooth inner surface, to the size of a whole classroom. And this is really important because now consider that this entire floor of the classroom is made up of epithelial cells that are all absorbing at the same time. If you got all those cells absorbing at the same time, we can absorb things pretty darn quick. All right. Now, another thing that I wanted to mention is that we need to talk about kind of the big picture of how these nutrients are digested before they get to this site right next to the epithelium of these villi. So here you got the stomach drawn in green. This represents all the food that's being converted into chyme as the stomach kind of grinds up all that food and liquefies it. Now, once that food has been turned into this liquid chyme, with each contraction of the stomach, a little bit of that chyme is going to be squirted into the first part of the small intestine, which is the duodenum. The first problem we have is that this chyme is super acidic. And the duodenum doesn't have that protective mucus like the stomach does. So anything that's really acidic like that is going to damage the inner lining of the small intestine. Thankfully, we've got the pancreas. This is an organ that lies just beneath the stomach, kind of behind and beneath the stomach. It's going to secrete a bunch of bicarbonate ions, which is a base, into the duodenum, which is going to neutralize that chyme. So right off the bat, it gets neutralized. In addition, the pancreas is going to secrete a whole bunch of enzymes which are going to help us digest proteins, fats, and carbs. It's going to secrete pancreatic proteases, which are going to be geared towards proteins, pancreatic lipases, geared towards digesting fats, and pancreatic amylases, geared towards digesting carbs, right? So all these enzymes get secreted into the duodenum where they mix with that chyme, and they continue to break it down. By the time these nutrients entered into the duodenum, let's say we started out with a big old starch in the mouth, right, when we ingested a piece of bread. Salivary amylases in the oral cavity are going to start to break that down into smaller chunks of polysaccharides. Then it gets down to the stomach. You're going to have hydrochloric acid, which continues to break it down into smaller chunks of polysaccharides, but it's nowhere near being small enough because remember, it's got to be a monosaccharide in order for it to get absorbed. It then mixes with the enzymes that are secreted by the pancreas. Those enzymes are going to continue to break it down until those sugars are pretty darn small. It's going to break them down to about the size of a disaccharide, 2 monosaccharides stuck together. Okay, but that's not big enough, or that's not small enough to absorb. This is where the epithelium of the villi come in. Locked inside the epithelium or the cell membrane of these epithelial cells are these little yellow specks. Those represent brush border enzymes, which are going to do the final job of breaking this disaccharide into 2 monosaccharides. Now, these enzymes are locked inside the cell membrane, so they don't go anywhere, but they break those monosaccharides apart in the perfect location, because once they are finally broken down into monosaccharides, they're right next to the epithelium that's going to absorb those nutrients. And that's exactly what's going to happen. These monosaccharides are going to be absorbed into those epithelial cells through active transport. Once they enter into the epithelial cells, then they are going to diffuse into the underlying blood vessels through facilitated diffusion. Once they enter into these capillaries that are located beneath the epithelium of the villus, then those capillaries are going to take it to the liver for processing, and absorption is pretty much complete. The same process happens for proteins. By the time proteins get close to the epithelium of the villus, right here at this level, they're small chains of amino acids, maybe like 2 or 3 amino acids locked together. You're going to have different brush border enzymes that are going to break those small chains of amino acids down into single amino acids, where they're absorbed through the same mechanism, through active transport into this epithelial cell, where they then diffuse into the capillary through facilitated diffusion. Then taken to the liver for processing. Now, fats are different. Fats create a new problem because fats don't dissolve in water. And all of our enzymes that are geared for digesting fats are water soluble, so they're not really going to work. So in order for us to digest fats, we need to somehow figure out a way to dissolve them. And that's where the gallbladder and bile comes in. So, think about when you're doing dishes, let's say you're trying to clean a greasy pan, you're going to need soap in order to clean up that pan. And what soap is, it consists of tiny little chemicals that are attracted to both water and oils or fats. And because they're attracted to both of them, it's going to allow you to break apart those big globs of fat on the pan into millions of tiny little pieces, which you can wash down the drain. It's essentially made the fats water soluble in a kind of a roundabout way. Well, that's what bile is. The liver is going to create this substance, or this liquid called bile. It consists of bile salts that really act like a soap. The bile is stored in the gallbladder just beneath the liver, and when we consume fat, the gallbladder is going to secrete this bile into the duodenum using the same duct that the pancreas uses. This is actually the hepatopancreatic duct. What these bile salts do is that they're going to allow these big globs of fat that we have consumed to break apart into millions and millions of little tiny little pieces. That process is called emulsification. Once they're broken apart into all those tiny little pieces, they're water soluble, and that allows for the pancreatic lipases to digest these fat molecules. It's going to digest these large fats into tiny little fatty acids, which ultimately can be absorbed directly through simple diffusion into our lymphatic system. So bile is really crucial for digesting fats. If you get your gallbladder removed, that's obviously going to limit your ability to produce bile or secrete bile into your duodenum, and it's going to prevent you from digesting those types of nutrients. So if you eat some really fatty foods, if you don't have a gallbladder, most likely you're just going to pass that fat straight through your stool, right? And that's about it. Thank you.